In the second part of this chapter, we're going to have to take a closer look at chromosomes. What are they and what do they do? And the reason why is because later on we're going to talk about chromosomes quite a bit, and we need to have a basic understanding of how they work and what they are. Now, eukaryotic chromosomes are made up of DNA, of course, that's the genetic material, and the DNA is assisted by a variety of proteins called histone proteins. These histone proteins allow the DNA strands to not get completely entangled, to assemble and disassemble as they need. And once you put them together, so once you take the DNA and wrap it around some of these histone proteins, you call this assemblage chromatin. And in this slide, you see that the chromatin is stained. It's stained as sort of a purple, a light purple, and then when it gets all wrapped up, it turns into these dark purple chromosomes. Now, during interphase, which is when the DNA needs to do things, that's when the chromosomes are unwound. So the DNA is going to be openly available. There's a lot of activity possible. The information on the DNA can be read, and as a consequence, the chromosomes need to be freely available and the DNA needs to be unwound. However, when you need the DNA to replicate before mitosis, it's important that it becomes organized. So what you're going to have to do is you're going to have to wind it up like a ball of string, make sure it's organized, make sure that all the pieces are there, because only if all the pieces are there can you actually make a true copy. And that's ultimately what you need in order to preserve all this information. So the chromosomes are kind of like our disks or our files where all this important information is assembled and kept. Now, if you look at how DNA is coiled, it's rather amazing because you end up with this huge, long molecule, up to 6 feet, it says, to be packaged into this tiny, tiny nucleus. Now, when we worked in the lab, we saw how small our own cheek cell nuclei were. They were the little blue stained particles inside of our cheek cells, and inside of each one of them, you have up to 6 feet of DNA, coiled and folded and packaged. That's no mean feat. So if you look carefully at what happens here, you will see, first of all, up here on the right, that's the actual DNA. It's a double helix, and we talked about this in chapter 3. Now this strand of DNA is then assembled with the help of these histone proteins. So there's 1, 2, 3, 4, 5, 6, 7, 8 of these histones, 8 of the histone proteins. They help the DNA wrap itself around, and as a consequence of this wrapping, the DNA makes a nice, neat little nucleosome. That's what these are called. And if you look at this under an electron microscope, you actually see these as beads on a string. So here's a nucleosome, here's another one, there's another one, and so on. And we can actually see this under the microscope. But that's not all. The DNA gets folded into these nucleosomes, and then it forms these helical fibers. And then when these helical fibers are working themselves, they can be packaged and assembled into something called looped domains. And that again gets looped, and you end up seeing these very fine strands of a chromosome. And so this is the chromosome. This is a chromosome that has been duplicated. And so you can see on the one side here, on the top, that's going to be the information 1. So that's the first one of these. And then down here, you're going to have the second one. They're attached in the middle by a thing called a centromere. So that's how the chromosomes are put together in our bodies. Now, it's very interesting to see how all this comes together. And in the homework, there's a really cool movie that helps us understand this. Now, in order for us to move this into mitosis, we're going to have to make sure, as I mentioned, that the genetic material is replicated. And if you do this properly, then you're going to end up 2 copies of the DNA. This, of course, happens during the S phase of the cell cycle. And these 2 copies are then called sister chromatids. The sister chromatids are joined at the waist of the chromosome, and that is what we saw in the previous slide as the centromere. So that's the waist of the chromosome. And then during mitosis, the sister chromatids are separated, moved apart, and distributed to different daughter cells. Now I want to point out something that's very important so that you understand. This here is one chromosome. So the one chromosome contains all the genetic information that is needed. So this is one single strand of DNA, and it's wrapped around and packaged the way we showed earlier. After it replicates, this is still one chromosome. The difference now is that it no longer has only one piece of information, but now this information exists twice. So this is one piece of information, and that's equivalent to that, and this is the copy. Now this is a really good copy because we obviously need to make sure that the next cell has the exact correct information that it needs. And so that's how we distribute the chromosome material to the daughter cells. So the sister chromatids are basically the exact same piece of information, and those are each distributed to the different daughter cells. And that's how chromosomes work.